Dear children, welcome to the biology online classes. So, we were studying chapter 2, reproduction plans, before the pre-midterm examination. Okay, so we have seen till pollen pistil interaction, isn't it? I told all the type of pollen grains cannot grow on the stigma of any plant. Okay, if the pollen grain is of the correct type, okay, uh, that is if it is from the same species, then, or if it is compatible, okay, then only what will happen, the pollen pistil interaction will be successful. Okay, so if the pollen pistil interaction, what is happening in this pollen grains and the chemicals in the stigma, there will be a continuous conversation, means there will be a lot of chemical reaction, and if it is what compatible, understood, then what will happen, that will lead to the germination of the pollen grains, understood. So that all can be called as post pollination events. Okay, that is, the pollen will be accepted by the pistil, and then what will happen, that will lead to the germination of pollen grain. So pollen grain, you can see, that will start its growing, okay, from the stigma to the style, and then it will reach to the where, to the ovule, isn't it? So do you remember the parts of a carpel? Stigma, style, and ovary. Ovary will have what? Ovule. Okay, ovule or ovules, understood. So it will come down, and then what, it will go enter into the where, to the ovule. Okay, and this ovule, this what to say, this growth, that when the pollen tube grows, the entire content of the pollen grain also will enter into that, and through what it germinate, through the germ pores, isn't it? Germ pores will be there, and through the germ pores only pollen grain, pollen tube comes out, and the content of the pollen grain also will be moving to the pollen tube. Now it will reach to the ovule, I said, isn't it? And during this journey, the pollen grains, okay, which are shed at two-celled condition, will have two cells. What are they? Generative cells and the vegetative cell. And the generative cell, okay, as this grows down, it will form two male gametes. Understood? Two male gametes will be there, and this plants which shed their pollen grains in the three-celled condition, pollen tubes already carry the two male gametes. Understood? Then after reaching to the ovary, so do you remember, the ovary is having what? Micropyle structure of ovary, ovule, you remember? Okay, it reaches to the ovary and then enters into the ovule. Ovule structure, do you remember? Okay, it is having micropyle, integuments. Okay, so through the micropyle end, what is micropyle? That is the region where there is no integuments. Through the micropyle, it enters. Okay, when it reaches to the ovary, it enters to the ovule through the micropyle. Okay, and you have learned that in the micropylar region, egg apparatus is kept, isn't it? What are the parts of egg apparatus? There will be two synergids, okay, and a central egg, isn't it? Synergids will be there and egg will be there. So synergids are having filiform apparatus, okay, which helps in the guiding the male gametes to the egg. Okay, you can see here, these are the filiform apparatus where you can find in the synergids. Okay, and they guide the male gametes towards the egg. Egg is the cell, isn't it, where what happened, means egg is a haploid cell where it fuses with the male gamete and form the zygote, isn't it? And the other male gamete, how many male gametes are produced? Two. So one will fuse with this egg of the egg apparatus found in the ovule and form the zygote, and the other one will, you can see here, polar nuclei are there. I have explained all this when we were studying the embryo sac, isn't it? So in a central cell, in a cell called central cell, there are two nuclei. They fuse to form a diploid cell. Understood? And now one more male gamete will come and fuse with this, isn't it? And this is becoming what, a triploid cell, and this is known as, okay, after fusion of the polar nuclei and the male gamete, it is known as primary endosperm nucleus. What is it? Primary endosperm nucleus. And what will be the ploidy of that? It will be triploid. Okay, and it is used for what? It is used for the means growth of endosperm, the nutritive tissue. Is it clear? So this is what happens after the pollen grain germination happens. So till this pollen growth, okay, pollen tube growth, we can call it as what? Pollen pistil interaction. Understood? Pollen pistil interaction. If the pollen grain is of the the correct type, then only what will happen? Post pollination process will continue. Otherwise, what will happen? The pistil rejects the pollen, okay, and it prevents the pollen germination. Understood? So in this way, pollination means the fertilization will not happen. Okay, so this is due to a continuous dialogue between pollen grain and the pistil. Okay, there are lot of chemicals involved in this, and it is what now scientists were able to identify these components, what will lead to the rejection, okay, recognition, or after that rejection or acceptance. So this is very useful information, okay, because sometimes what incompatible pollination means, usually pollination we cannot say, the pollen pistil interaction, if it is not what compatible, what will happen? Simply pollen grain cannot grow. So in different stages only this pollen grain germination will be stopped. So if we have clear knowledge of all this, we can what help the plant hybridizers, okay, those who are doing this plant hybridization technique, to understand more about this, and they can even make it possible to to happen the pollination between two incompatible species, okay, so that we can produce the super varieties, isn't it? Varieties which are not common. Understood? So in this way, pollen pistil interaction is very important, and its knowledge is also very important, so that we can understand, okay, how this pollen pistil interaction happens, and why, if it is not successful, what are the reasons, and how we can overcome this, so that the what the farmers, okay, or the experts of agricultural or plant breeding techniques, okay, those who are doing all this can understand this, and can make it possible to to make the pollination successful. Is it clear? So if the pollination is successful, what will happen? I told already, isn't it? The two male gametes will be there, and that will come through the pollen tube and enters into the ovule. And in the ovule, okay, near to the micropylar end, the egg apparatus is there, that will, one of that will fuse with the egg and form this diploid cell, that is known as the zygote, and the other male gamete will fuse towards that will fuse with the polar nuclei and form the primary endosperm nucleus, PEN. And this, okay, as there are three haploid cells. What are the three haploid cells? Two polar nuclei, and then what? The male gamete, isn't it? Is involved in the formation of primary endosperm nucleus. It is known as triple fusion. Okay, and two sets of or two types of fusion is happening. That is synchrony and triple fusion. Okay, that two are taking place in the embryo sac, and because of that, it is known as double fertilization. Okay, and I told the central cell, okay, that after triple fusion they will become what? The primary endosperm cell, okay, and they develop into the endosperm that gives nourishment to the embryo. So this is happening in the what? Post fertilization means after fertilization, these are the things happens. Okay, so after that what happen again? Post fertilization events, that is actually up to this fertilization happens, and after that seed formation, endosperm formation, etc. takes place. That I'll tell you after another topic, that is what plant breeding technique. Okay, plant breeding technique. So I told you many of the pollen grains, isn't it, will not germinate on the stigma of any flower. Okay, there will be a successful interaction between them. Okay, so different plant breeding technique are there, and in that one is artificial hybridization. Okay, so artificial hybridization means between two different parents. Okay, if the crossing is done between two different parents, it is known as what? Artificial hybridization. So we can what? Cross different species or different genus also. Okay, plants belonging to different genera also. Understood? So that we can get some super varieties. Isn't it? Super varieties means which has the qualities of both the parents. Parents will be different. Okay, they may belong to different species, or they may belong to different genus. Understood? So this, so that we can get what? Super varieties. So in this, what we have to use, or what we have to do is, we have to collect the desired pollen grains, and that should be used for the pollination. Okay, and the stigma should be protected from the contamination. Contamination means from unwanted pollen grains. Okay, otherwise if the stigma is open, any pollen grain can what stick onto it, and that can affect our result. Okay, so for this, the major thing that is to be noticed in artificial hybridization is what? Collection of desired pollen grains, and the what the protection of the stigma. Understood? So for this, if the female parents are bisexual, okay, we cannot do this, isn't it? Because the anther is there in the flower itself, so pollen grain of the same flower may fall on the stigma. So it is very very important to remove the anthers from the flower before the anther dehiscence happens. Okay, so this step is known as what? Emasculation. So what is emasculation? The removal of, okay, anthers. Okay, removal of anthers from the flower bud before the anther dehisces. Okay, is known as what? Emasculation. And then what we have to do, if the emasculated, what is emasculation? The removal of anther. Okay, so the flower will have now what only? The anther, the stigma only will be there. Understood? So this stigma may get contaminated by any other pollen grains, unwanted pollen grains. So that should be protected from other foreign the pollen grains. So for that we can just cover the stigma. Okay, and this process is known as bagging. Understood? So bagging, why it is necessary? To protect the stigma from other unwanted pollen grains. Okay, so when the stigma are bagged, okay, and when the stigma, okay, they get matured, okay, to receive the pollen grains, the pollen grains from other what the desired plants, okay, which we are going to do. So from that plants we have to collect the pollen grains. Okay, and then it should be dusted on the stigma. And then what will happen after the pollination is successfully done? Again it should be rebagged, so that what will happen? The fruits will be formed, the seeds will be formed, etc. Understood? So if bisexual flowers are there in artificial hybridization, then it is very necessary to remove the anthers. So this process is known as what? Emasculation. After emasculation, what should be done? Bagging should be done. What do you mean by bagging? In bagging, we cover the stigma with the help of a bag, or polythene bags can be used. Okay, and it should be tied. And when the stigma get means the flower get matured, when it is having the receptivity, we should dust the pollen grains from the desired plant. Okay, and then again it should be rebagged. Understood? Then we can collect the seeds for the sowing. That plant will be having superior qualities in it, because we are hybridizing. We are selecting two different parents, and from them one will be considered as the male parent. Which one will be considered as the male parent? From which we have collected the pollen grains, and the other will be what the considered as the female, where we are using the stigma. Understood? So like this we can do the artificial hybridization. And if the female parents are having unisexual flowers, is there any need of emasculation? No, isn't it? So like this there is no need of that, and means emasculation. And what we have to do, the female flower should be bagged before the flower open. Otherwise what will happen, there will be chance of other contamination of the stigma with other pollen grains. Okay, and when the stigma becomes receptive, pollination will be carried out using the desired pollen, and then it should be rebagged. So this artificial hybridization was asked once. Okay, different steps of artificial hybridization. So what are they? First, selection of the parents, isn't it? We should select the parents. One should be considered as the male and the other should be considered as the female. So how we will take consider the parent as the male? So from which we are collecting the pollen grains, that will be considered as the male, and the other will be to which we are dusting the pollen grain, okay, after emasculation or after bagging, that is known as the female. Okay, that will be considered as the female. Then second is what? If it is bisexual flowers, what we have to do? We have to remove the anthers. What is that step known as? Emasculation, isn't it? After emasculation, bagging. What is bagging? Covering of the stigma, so that there is no contamination of the stigma with the other pollen grains. Okay, then next is what? Pollination, isn't it? We have to collect the pollen grains from the what the flower which is we thinking to make it as the male. Okay, so from that we have to collect the pollen grains and dust it on the stigma. Understood? Then again after pollination we have to rebag it. Okay, label it. Label means date of pollination, all that can be written. Understood? The variety which is used, all that can be shown in the label. So this is what the procedure of artificial hybridization. So if anyone is doing artificial hybridization in this way only, it will be done. Okay, and if the female parent is producing unisexual flowers, then what will be the change? There is no need of emasculation. Then the flowers will be bagged, okay, before the flower open. This also you notice because it can be asked. Okay, if the flowers are unisexual, how artificial hybridization can be done. So is it clear? So if the females are if the flowers are unisexual, okay, then there is no need of emasculation. Then the female flower buds, okay, before they get opened, should be covered with a bag. Okay, and when the stigma become receptive, pollination is carried out during using the desired pollen. Okay, and then it should be rebagged. Okay, so this also was asked once for 5 marks. Okay, artificial hybridization. So different steps you have to remember in this. Understood? What are they? First is selection of parents, that is not given separately here, but it is understood, isn't it? We have to select one plant as the male and the other as the female. Okay, and then what we have to do? If the flowers are bisexual, okay, in the female flowers means the flowers which we are going to select as the female flowers. If they are bisexual, then what is needed? Emasculation, isn't it? And after that bagging, and then pollination. Okay, artificial pollination, and then what will happen? Again it should be rebagged. Okay, so and if the flowers are unisexual, female flowers are unisexual, then what is the advantage? There is no need of any emasculation. So emasculation, how we will do, children? With the help of forceps we can remove. Okay, or sometimes what we can use some cold water treatment so that this, or hot water treatment so that the stamens will get inactivated. Okay, or else we can use what some genetic technologies so that the flowers will not be having the male part. Understood? So by different ways emasculation can be done. Okay, so emasculation usually with the help of what? A scissor or forceps. Okay, forceps, with the forceps we can manually remove the anther. Understood? So what we have studied today is the after pollen interaction, pollen pistil interaction. Okay, what is pollen pistil interaction? Pollination to the growth of pollen tube to the entry of pollen tube to the ovule. Understood? So this much, all these events are coming under pollen pistil interaction. So if pollen pistil interaction is successful, then it will lead to what? The formation of zygote, and then what? The primary endosperm nucleus. I told this. Then another topic I have told, that is artificial what? Hybridization. That is actually a different topic. Okay, artificial hybridization is not coming under any of this process. It's another topic. As we have studied pollen pistil interaction, we have studied this also in this. Okay, so hope this much is very clear. Let's continue in the next class. Thank you.